Here is the template DNA strand before it goes through the replication process. Each strand is led by a carbon 3' or a 5'. The DNA strand is moving closer to the replication fork. In the replication fork is the protein helicase. Helicase is an enzyme that speeds up the reaction and splits the DNA strand apart. It breaks through the weak hydrogen bonds that are holding the nitrogenous bases together, adenine with thymine and guanine with cytosine. The DNA strand continues to move through the replication fork, separating from itself to create single-strand binding proteins. In this case, the leading strand is on top and the lagging on bottom. The leading strand has continuous repetitions towards the fork, and the lagging strand contains breaks while moving away from the fork. The enzyme primase preps the DNA with a primer so that the DNA polymerase will eventually know where to start. Now enters the DNA polymerase. Its function is to replicate DNA molecules to form a completely identical DNA strand. It will move from prime to prime, building off of the 3' end in the 5' direction. This is what creates leading and lagging strands, because the DNA polymerase cannot continuously build on the lagging strand. It needs to continue to be primed so that the DNA polymerase knows where to restart after each break. Free nucleotides are used to fill the space between the Okazaki fragments in the lagging strand. Ligase is another enzyme that helps fill the gaps in the lagging strand. It takes care of the extra gaps and seals the Okazaki fragments together. The DNA replication process continues as the new strands form with coinciding base pairs. DNA replication is semi-conservative because each new DNA strand contains an original strand and a newly made one. It also happens in an anti-parallel fashion because the strands are complementary and don't go in the same direction.